Hello, this is Mr. Merrier with Noteworthy Science. Today we're going to be doing subtopic C3.2 Defense Against Disease. Under the curriculum matrix, this falls under theme C, Interactions and Interdependence, and under the level of organization of 3, Organisms. Some guiding questions to keep in the back of your mind as we're learning this subtopic is, how do body systems recognize pathogens and how do they fight them? And how do they fight infections? And what factors influence the incidence of disease in populations? So really relating to epidemiology. This subtopic is a standard level only subtopic, meaning that there are no additional higher level learning outcomes. And in this one, there are 18 different learning outcomes that we're going to be going over today. Let's begin. The major role of the immune system is to identify and destroy pathogens. And so a pathogen is a fairly general term, meaning any disease-causing organism. And organism here is used somewhat loosely because it's not just living things that can cause disease, it's also agents such as viruses or even prions. And so the idea is that if an agent, if a thing, if an organism is causing disease, then it can be classified as a pathogen. A disease is really any abnormal condition that has a negative impact on either the anatomy or physiology or functioning of an organism. And if something is going to be classified as a disease, then it typically comes with associated specific signs and symptoms. So if someone has, let's say, COVID-19 as a disease, does this person, let's say, have shortness of breath? Has this person lost some taste? Does this person have an elevated fever? Things like that. These are symptoms that are associated with a particular disease. So it'd be unlikely that you and I have the same disease but have none of the same signs and symptoms. Of course, it doesn't mean that you have to express all the symptoms of a disease, but they are typically marked by very classic things, and that's what allows diagnostics to happen. That's how a clinician, that's how a doctor can be able to diagnose, do you have this disease, yes or no, particularly. And we typically have fairly robust definitions for how to classify something as this particular disease or not. A pathogen can cause disease by infecting a host organism. Right? And that means that they're going to be able to enter the host, and what's going to happen is they're going to start to grow and multiply. And in so doing, they're going to start causing damage. That either means that they're going to maybe potentially destroy your cells, or it could even means that they're just taking a lot of nutrients that otherwise would have went to your cells. Either way, they're causing problems within your own metabolism, within your own physiology. Now, if that pathogen can be transferred from one host to another host, then we would say that this pathogen now is communicable, or that disease is infectious. It means that this disease can be spread from one person to another person. Keep in mind that there are diseases that are not pathogen caused, like cancer, for instance. Cancer is not transmissible. If I have lung cancer, I cannot transmit lung cancer to you. The way in which a pathogen is spread from one host to another host obviously varies significantly, and that will largely depend on the pathogen itself. So I want to just go through a few examples of different pathogens and their respective diseases. It is not a learning outcome that you know these, but I think it is worth me discussing the differences between the pathogen and the disease, because I think a lot of people confuse these two. So SARS-CoV-2 would be the name of the virus, and we actually specifically looked at this virus at the HL level, and that can cause COVID-19. Or the virus, HIV, can cause the disease of AIDS, right? The rhinovirus causes the common cold. A bacterial disease, as their name implies, are caused by bacteria, not viruses. And so some serious ones, or some famous ones, would be like pneumonia, which would be the infection of the lungs, hence pneumo, right? You have things like the bubonic plague, you have a UTI, or urinary tract infection, tuberculosis, strep throat. I would say there is a really big misconception between viral and bacterial causes of disease. In fact, I think still to this day, when people get sick, they will attribute this incorrectly to bacteria. And the reason that I suspect this is because people still take antibiotics for things like the flu or things like the cold. But an antibiotic, as we'll look at more in this topic, and we've already explored in other topics, target bacteria. And if something is viral in nature, this is going to be entirely ineffective and potentially even cause further damage because they can be actually harming beneficial bacteria in the host. So understanding what your disease is caused by is crucial to being able to fight it and to be able to take the correct medication. So if you want to fight a viral disease, then you would take an antiviral medication. You would not take an antibiotic. The other types of pathogens that you may or may not have been aware of would be protists. So protists are single-celled eukaryotic organisms. And you actually probably have heard of some of these diseases before, but you may not have known that they were protist in nature. So malaria, I think a lot of people would incorrectly attribute malaria to mosquitoes. Mosquitoes would be considered the vector of the disease, meaning they are the ones that are actually carrying the pathogen. In this case, they are carrying the protist that will ultimately cause malaria in a potential host. And the same thing could be true for something like rats being blamed for the bubonic plague. Well, the rats themselves aren't causing the bubonic plague. That's a bacteria, but they are the vectors in how it was. And in fact, even the rats, you could argue, are not the true vectors. It's the fleas on the rats that were the vectors of the bacteria that caused the bubonic plague. So don't confuse a vector for a disease for the pathogen causing of the disease. Giardia is actually another fairly well-known one, and this is actually, if you ever were to drink, let's say, freshwater sources that are maybe contaminated, they may be contaminated with this. So you could get this kind of a disease from drinking, let's say, unclean water. And then finally, another disease that you may have had before in your life would be maybe something called athlete's foot or a fungal infection. Common ways, everyday ways that you can get something like this would be like walking barefoot in like public restrooms or in showers or things like that. Because fungi tend to aggregate in cold, dark, damp areas. And that's what allows them to actually grow on the surface of things. What are you supposed to get out of this? I think the main idea is that, one, there are a fairly large variety of different pathogens that can cause disease. We're specifically talking about human diseases, and we don't really know of any archaea that cause disease in humans. So that's why, if you're wondering why they were left out, that's where we're leaving that out here. So there's actually two really interesting case studies that I want to explore now through the nature of science learning outcome. And so what epidemiology is, it's the study of what is causing disease, where is the disease actually occurring, how is the disease actually being distributed. So it's really looking at it from a population or a macro scale. Where are the outbreaks happening? What are the cause of the outbreaks? How can we try to mitigate the spread of that disease so that we can contain it potentially in this area before allowing it to go further? So epidemiology is, as you hopefully at this stage understand why it's so important and why keeping track of diseases and how they spread is of paramount importance, so that we're prepared if anything were to happen, let's say, again. This first case study is one that you may have heard of before because there's a university named after him in Hungary, and that is Semmelweis. Pretty amazing story. So in the 1800s in Vienna, the germ theory of disease was not really recognized yet, meaning that the idea that germs are spreading disease from person to person. So that was not really a well-known or well-accepted idea at this area at this time. Semmelweis was a physician who worked in maternity wards, and so in wards where he was helping deliver babies. And what he noticed is that there was a fairly large mortality rate in women after they've given birth due to this fever, or the colloquial name would be this childbed fever. And what he noticed was that it's not just that these women were dying of this fever, is that the hospitals had higher mortality rates than if women were giving birth, let's say at home with like midwives, or even on the street. Street births had a lower mortality due to this than hospitals did. And that was obviously a fairly big alarm bell to him anyways. How are hospitals having the higher mortality rates compared to at-home deliveries or even deliveries on the street? What is going on here? And so what he hypothesized was that it was actually the doctors delivering, the obstetricians, that were actually spreading disease to the mothers. And the reason for this, and this may sound weird or kind of wild in our day, but the same doctors that were doing autopsies would come from an autopsy and then help deliver a child. Again, there was no idea of disinfecting hands. That was not really a thing. It was not common practice. It, in fact, wasn't done at all. So imagine the kind of bacteria that may be present on a corpse that are on still a doctor's hands that are then being used to help deliver a child when they're going to have potentially direct contact with the mother. So he started, this idea was not believed. People did not really believe him, but his own clinic, he actually started doing almost his own study, and he urged all the nurses, all the doctors to sterilize or disinfect their hands prior to seeing patients. And then what he saw was that mortality rates started to massively decline. This was not recognized, I believe, not until far, far later, potentially after his death. That he was indeed correct in this. And this is why even to this day, there are very, 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 very strong protocols for disinfecting and going into any surgeries or anything like that. And that is to mitigate or prevent the spread. Now, what should you get out of this story? It's really, he made observations. He saw, why is disease much higher in this scenario versus this scenario? Right? He didn't come up with this idea because he physically was able to observe the pathogens. That had nothing to do with it. It was inconsequential. He was just saying that, listen, the incidence of disease is much higher in this area than it is in this area. Why might that be the case? And that's really the core of epidemiology. Why is disease spreading here but not here? What's causing this area to be more infected than this area? What's happening? And that's really what epidemiology is. And another example that we're going to look at, which is Dr. Snow, is maybe sometimes referred to as the father of epidemiology. And this is another quite famous case study. In the 1800s in London, there was a really large cholera outbreak. And if anyone here knows what cholera is, cholera is a disease that is caused typically from consuming infected water. Usually when fecal matter enters the water, and then you're drinking that water, that causes cholera. It's actually still, unfortunately, quite prevalent in a lot of underdeveloped countries around the world. And the reason why this is so, what this disease causes is prolonged diarrhea. And that's problematic because it basically leads to dehydration. And that's the big issue here. So there was a big cholera outbreak in London. An outbreak suggests that a huge number of infected people over a short period of time in a small area. So what he did, and again, which was novel at the time, is he started conducting interviews and he started to collect evidence. Which people are being infected? Where do they live? Where do they obtain their water? Where do they get their food? And he started to make patterns. And what he did is he literally created a map of London and plotted where the case studies of cholera were. And by doing this, he was able to pinpoint all the cases, or majority of the cases, seemed to stem around this one water pump. And he hypothesized that this pump was contaminated and was the source of cholera in the first place. And so he urged the city council to basically turn off this water pump so that residents could get their water elsewhere, and cholera levels ended up dropping as a consequence. This is why it's often referred to as the father of epidemiology, because this is effectively what epidemiology is. Yet again, he looked for patterns. Where are the outbreaks? Where are the case studies high? Where are they low? Why might this be the case? Start making patterns. And again, this is what with the advent of the internet and the advent of instant communication, epidemiology now can be done on a global scale. This is why it's so imperative to report infections, report studies, report deaths. Getting this information to organizations like, let's say, the CDC would allow them to pinpoint, are cases above, let's say, normal, or are they getting more and more and more frequent? What's happening? What could be the cause of this? And that's what epidemiology is. So we can't ever eliminate pathogens in the world. They're just a thing. So the idea is, how have our bodies evolved to be able to deal with this continual threat? There's a series of, let's say, mechanisms in place to try to stop an infection. And the first one, it's almost like the best offense is a good defense, is kind of what this is, a way that you can think about this, which is, what's easier than having to fight an infection? Preventing the infection from occurring in the first place. And that's where primary defense comes in. Your primary defense in a body is really a barrier. What physical and chemical barriers does an organism have to prevent a pathogen from even entering in the first place? That's what these are. So this is just like akin to a wall, right? It's a wall in a city. I don't want anything coming in. That's how you can think about physical and chemical barriers here. So the first primary defense that you may not have considered is your skin. I think a lot of people are just like, well, your skin is the thing holding you together. Well, sure, I mean, that's true, but the skin has a lot of other functions. And a big function that your skin has is as a primary defense to any potential pathogens. And it's a very good defense to any pathogens. So let's talk about why the skin is such an effective barrier. Note that you don't have to be able to label the layers of the skin. That's not necessary. But I'm going to be explaining how the skin actually functions as a really effective barrier here. If we go deep into the skin layers here, there's a lot of mitosis happening here, where new cells are constantly growing, dividing. As they are growing, dividing, they migrate upwards towards the surface. As they migrate towards the surface, they are now going to compress and basically die. As your cells come up towards the surface, they will then start to massively compress, and then they'll actually die. And that death actually allows them to compress even more. And so now you get basically this fusion of a bunch of cells very close together. And on top of that, if I zoom into this top layer now, so here are these dead skin cells. They are connected together with a very hard protein called keratin. And you've likely heard of this protein before. Keratin is also found in your hair, in your nails. It's a very tough protein. So why is this already a really good barrier? Well, if I'm a pathogen trying to come in, not only do I have a wave of cells pushing against me, but I have a huge amount of layers that is super hard because of keratin, and they're not even cells that are alive that I can infect. They're dead. It's just a huge compression of a bunch of cells at the top. So you have this never-ending almost wave of cells coming up this way, making it really nearly impossible for a pathogen to ever make it all the way down. It's very difficult. And this might be surprising to you, but you are shedding skin cells right now. You're shedding skin cells all the time. And that's on purpose. That's because it makes it very difficult for pathogens to actually penetrate that layer. On top of that, your skin also has some chemical deterrents. One of those being acid and sweat. So your skin, through sweat glands, actually secretes sweat on the surface. Now that's not just a thermoregulatory mechanism, it's salty, right? There's a lot of salt in your sweat, and that causes a fairly inhospitable environment for a lot of microorganisms to live. Secondly, your skin also secretes acid. So the surface of your skin is actually slightly acidic, again, really making it inopportune for microorganisms to reside and multiply there. And the last kind of thing that your skin does, which may really surprise you, is that your skin is already home to native bacteria. In fact, some estimates put the native bacteria on your skin to be roughly 1 million organisms per centimeter squared. So you already naturally have bacteria living on your skin. Now, this does not seem like a very good barrier, but it is. Because if a new pathogen is hoping to colonize here, they have to compete. They have to compete with the bacteria that are already thriving on the surface of your skin. So that is actually the competition of your native bacteria here is actually a barrier in and of itself. So for all of these reasons, your skin provides a really good barrier to pathogen infection. The other major primary barrier that we have is in the way of mucus. So the skin is awesome, very, very difficult to penetrate. I'm not fully coated in skin, obviously. There are natural openings that you have that have to remain open. That means that anywhere where there is an opening in your body, this is a potential site of infection. So these would be things like your mouth, your nostrils, your eyes, your ears, your urethra, your anus. These are all natural openings. That means that they are all possible entry points for a pathogen. So your body obviously can't seal these up. So what they're going to do is they're instead going to line them with mucus. So a lot of these orifices secrete mucus. So mucus is a viscous fluid that actually contains other things. So first of all, it's viscous. Why is that relevant? Well, let's use my respiratory system as an example. I am constantly inhaling maybe small particles, maybe dust, maybe particulates, maybe viruses without my knowledge. They're way too small for me to see. But as they come into my respiratory tract is lined and constantly secreting mucus. They'll get trapped in that mucus, and then you have cilia lining your respiratory tract that will literally beat to move my mucus back out. Hence you blowing your nose, hence you sneezing, whatever. That's what's going on here. Your mucus has trapped things that shouldn't be in there, and then your cilia are literally pushing them out. Common misconception, can anybody tell me the difference between cilia and villi? So cilia are really just projections, almost like hair-like projections from the surface of a cell. And again, they're always going to be lining the inside part of a cavity of some kind, right? And their main function is movement of something above them. So areas in the body where you might find cilia is, like I just said, your respiratory tract. You would definitely see a lot of ciliated cells here trying to move mucus. You would also see them in the oviducts within females that are actually physically moving the oocyte all the way to the uterus, right? So these are all involved in movement. Villi or microvilli are foldings of a cell. So this is literally just your membrane folding to increase surface area. Where you would see villi and microvilli would be areas of exchange. It's where you want to exchange things a lot. Things like in the kidneys, things like in your small intestines. So do not confuse these two things. We definitely don't want exchange here. That's the opposite of what we want. We want to move things out. So this is effectively like crowd surfing. That's what this is. There are literally a bunch of things moving the mucus with all the stuff trapped inside of it out of the body. So your mucus is not just a good trap, it also acts as a chemical deterrent because it contains enzymes. And specifically, it contains a kind of enzyme called a lysozyme, which can actually kill bacteria. And so not only can your bacteria potentially be trapped in the mucus and then physically get escorted out, they could be killed in that mucus before even infecting in the first place. And areas that don't necessarily have mucus but still have something similar would be something like your tears. So the reason that we have tears in the first place is probably lubrication, and that is because your eyes are constantly moving. In fact, the Muscles controlling your eyes are one of the most active muscles in your entire body because they're moving literally all the time. So you need a lot of lubrication there. So that's why you have tears. But in addition to that, your tears also contain lysozymes. Your tears also contain enzymes because this is another potential site of infection, right? I think a lot of people intuitively maybe forget this. Let's say people that don't wash their hands often, which is a lot of people, and then they'll touch their eyes or they'll rub their face. So what you've just done is you move something that, yeah, if there's bacteria or something on my skin, maybe not a big deal, but I've now just moved that directly to my eye. That's a problem. So the idea is that any of these areas are potential sites of potential infection. So your body has two major ways to prevent them from coming in the first place, and that being the skin and mucous membranes. We have our primary defense. Now, let's imagine that defense is breached, i.e. a cut. So I have my blood vessels underneath, right? Now, normally, as we've already said, it's quite difficult for any pathogen to make its way down here. That's quite hard. But let's say I'm clumsy and I cut myself. If I cut myself, what I've basically done is create a direct path through all of those barriers. If you're literally bleeding, that means obviously a blood vessel has been ruptured, which means blood is now coming out. That is of course a problem because you don't want to lose blood in the first place. But even if the cut is really, really small and you're not probably going to die of blood loss, it is now a potential site of infection. So depending on what cut you, if let's say what cut you was a knife that's never been cleaned, or a nail, or something that's rusty, or a pretty dirty piece of equipment, these equipment pieces may already contain pathogens on their surface. You've now introduced a full pathway all the way into your system. So this is now a big problem for your body. What is your body going to do? And this is what the next stage that we're going to look at. This is the clotting process. So let's discuss the blood clotting, sometimes referred to as a cascade. So step one, your cells have been damaged. You are now losing blood out of your blood vessels and out into, let's say, through your skin. The first responders here are going to be these little cell fragments. They're not full cells themselves, but they're called platelets. Their true name is a thrombocyte. And again, cyte for cell, and thrombo because thrombosis is literally the nature of clotting something. So these platelets are already in your plasma. They are naturally dispersed in your plasma. So they're all over the place. When a cut happens, they're going to start to effectively aggregate at the cut site, and they're going to form a local plug. This is not enough to fully stop the bleeding, but they're almost like the first things that are going to be there. And your body is also going to do a secondary action, something that we looked at in B3.2. If I want to minimize blood loss to an area, what can I do? Vasoconstriction, your blood vessels can actually start to contract, meaning that the amount of blood flow to that area decreases now. So this is your body being like, okay, there's obviously an injury here and we're losing blood. Let's minimize any flow to that area. That's going to be kind of like the first step here. The next step, your platelets are now going to release these proteins called clotting factors that I've shown here in purple. Now, clotting factors have a really important role because they effectively are going to activate an enzyme. So also in your plasma, naturally dissolved, is the inactive enzyme called prothrombin. Now again, the names here I know may sound a little complex, but we can deduce the etymology here. Pro in this case means before, so it's an inactive version. Thrombin, or thrombosis, again, means clotting, and IN as a suffix is a protein. So we have the inactive clotting protein. So this enzyme is now going to become activated by clotting factors, forming thrombin. We now have activated thrombin. Great. What does thrombin do? Thrombin, as an enzyme, is going to catalyze a conversion. Also naturally in your blood plasma is a soluble protein called fibrinogen. This fibrinogen I showed here in dark green. So fibrinogen is a naturally plasma protein that is soluble. That means it dissolves in the plasma, no problem. In the presence of thrombin, it gets converted into fibrin. Now again, the word here should kind of already tell you what it's going to do. It is a fiber-like protein. All of these fibrin are going to now link together in a mesh, and they're now going to form an insoluble protein, which then will form over the wound. Now you might be like, how is placing a net over a hole gonna stop blood flow? How is a mesh gonna prevent blood loss? That mesh is gonna trap red blood cells. That's what forms the clot. This is what a scab is. When you form a scab over a wound, that's really just a collection of dead cells that have aggregated at the wound. And this is literally causing a temporary plug, right? Scabs aren't permanent. What a scab is almost like this is now under construction. Your body is now going to repair that. That's going to take time, potentially a few weeks. In that time, we want to make sure that we're not losing any more blood, and we want to make sure that there's no pathogens coming in. So this is also why, well, despite the fact that it maybe is satisfying to peel your scabs, you're kind of really annoying your immune system here. It's kind of like, oh, we have to restart the entire process all over again. Thank you very much. So as a summary, this is why this is referred to as the clotting cascade. A cascade being one thing leads to another, leads to another, leads to another. If you look at this clotting process, we have platelets release clotting factors. Clotting factors activate prothrombin to thrombin. Thrombin catalyzes the conversion of fibrinogen to fibrin. Fibrin causes a mesh which traps cells, forming a clot. Now that you understand this process, think back to B3.2 and coronary heart disease. If the interior of your coronary artery bursted, well, and that was already narrow, your body is going to treat that like a wound, so it's going to start the clotting process, and that's what can cause an occlusion. That's the issue. And especially, as we discussed as well, if you form that clot within the coronary artery, a piece of that can break off, right, that we call an embolus, and that can now travel elsewhere and cause an occlusion, let's say in the brain. Okay, so we discussed all the different kind of barriers and protections that your body has to try to stop pathogens from coming in. But sometimes it's inevitable. And even if the clotting process is really, really good, some pathogens are potentially going to enter. Or rather, some things that shouldn't be in your body are going to enter. And this is where the actual immune response is going to begin. So your immune system can be separated into two major categories. Something called the innate immune system and the adaptive immune system. So the innate immune system is the immune system that you're born with. And it's the immune system that is non-specific and does not rely on experience. This kind of response is basically targeting anything that should not be in your body. So it doesn't matter what it is, it's just, it shouldn't be there. It's non-self. So this response is just trying to get rid of anything that your body deems as should not be there or foreign. Your immune response can be carried out by a group of amazing different cells called white blood cells, or leukocytes, meaning white cell. So these white blood cells can be further partitioned into two major categories, those being your phagocytes and your lymphocytes. Now in this course, we're going to keep it broad and only deal with certain kinds of examples here, but keep in mind that the true immune system is much more complicated. So let's look at phagocytes. Phagocytes, or real examples of phagocytes, might be macrophages or dendritic cells. So these are cells that undergo phagocytosis, meaning cell eating, where they're able to engulf an organism via endocytosis and then break it down. So let's describe what would occur if a pathogen or something foreign has entered the body. What is your cell's primary response? What is the innate immune's role in this? So let's imagine that I had cut myself up here and then pathogens have entered here. So this would be the site of infection. Now, inevitably, whether that's bacteria, whether it's viruses, fungi, whatever, these intruders, so to speak, are going to start to cause damage. And they're going to do this in a number of ways. They're either going to do this by directly destroying cells, or it could be by their siphoning resources away, but either way, they're causing damage. They're causing problems within the body. So when this happens, your body cells are going to actually start to release chemicals called cytokines. And where the word cytokine comes from is actually cell movement. So these are almost chemicals that direct a cell to go towards a certain location. They initiate or promote cell movement in a directed way. So it's almost like I'm dialing 911. I'm dialing emergency services to get the help of other people. So these cells are in danger. They're releasing cytokines basically as a warning to the body. These cytokines can then travel throughout the bloodstream, and what's going to happen is there's going to be a gradient. Cytokines are going to be really high here, and cells that are very in tune to cytokines would be your leukocytes, or more specifically, your phagocytes. And they're going to start to move towards these cytokines. And typically, your white blood cells and phagocytes are restricted to be within your blood vessels. But their structure is in such a way that they can use what's called amoeboid movement and kind of move kind of like an amoeba and squeeze through the little pores that actually line your capillaries. And they can actually leave the actual blood vessels and get to the site of infection. So now they've reached the site of infection. What next? Well, now that they've reached the site of infection, how exactly do your cells determine what's foreign and what is not? Or another way of thinking about this is, how does a cell determine self from non-self? And the key behind all of this is going to be receptors. So your white blood cells, your phagocytes, have these receptors on the surface. But remember that these are not really that specific. These are your first responders. They're not really looking for a particular pathogen. So these receptors are looking for basically structures that wouldn't be in your own body. I think the true name for this could be what are called PAMPs, or pathogen-associated molecular patterns, or it's a fancy way of saying what are some molecules or structures that should not be present in, let's say, humans. And a good example of this would be like peptidoglycan. So bacteria use peptidoglycan in their cell walls. We do not use peptidoglycan. So if these receptors were to bind on and identify, whoa, this is peptidoglycan, it's a sign that this is foreign. This should not be here. So once it's able to successfully bind to one of these foreign antigens, it's going to cause them to become activated. Once they become activated, they're now going to undergo their namesake, phagocytosis. So they're actually going to manipulate their cell membrane to actually go around something called pseudopods. They're going to move around the actual pathogen and engulf it. And they're going to engulf it within themselves in a vesicle, which we can call a phagosome, meaning it's a vesicle that's derived from phagocytosis. Now, we haven't exactly done anything to this pathogen. We've really just enclosed it into space. So what? Well, the next thing that's going to happen is that lysosomes, which recall are organelles that contain very powerful hydrolytic enzymes, are going to now be initiated to move towards this phagosome and fuse. Once this lysosome has fused with this phagosome, we can now call this a phagolysosome. They're now fused together, but now all of these hydrolytic enzymes are now going to be released, and they're going to start to break down that pathogen at a molecular level. They're going to start breaking apart all kinds of components. So yes, they will ultimately eliminate most of the cell contents via exocytosis and eliminate it just like you would eliminate garbage. But they actually do something much, much cooler that you may not have been aware of. What they're going to do is another vesicle that contains a very special receptor is going to come into play. And this is called MHC2, or Major Histocompatibility Complex 2. This receptor is very, very key, because what's going to happen is that it's going to actually fuse with this phagolysosome, and what's going to happen is that it's going to take some of those antigens, some of those surface markers on the site of that pathogen, and it's going to bind to it. And this now bound MHC2 with an antigen is now going to fuse with the membrane surface, and it's going to present these antigens on the surface. In this way now, we can now call this cell an antigen presenting cell. What it's really doing is it's basically showing or demonstrating to the cell, hey, look what I just killed. It's almost like a trophy, so to speak. So instead of just taking the antigen and throwing it away, it's going to take an antigen, and if I am MHC class 2, I'm taking the antigen and presenting it this way, and I'm presenting it on the surface of my cell. Now, the reason that it's doing this is not really a flex. What it's doing is it's now going to become an integral part of the adaptive immune response. The adaptive immune response, unlike the innate immune response, has all to do with specificity, and it has all to do with experience. So this is not a system that you would actually have developed upon birth. This is one that you're going to develop consistently over your lifetime, and it's going to come from experience of different pathogens entering your body. And so this is very, very specific. So if the innate immune response is non-specific, basically targeting anything foreign, the adaptive immune response is very specific. It's targeting one particular pathogen, one particular thing that is deemed as foreign. So before we get into this process, let's first talk about where are these cells located. Now, the innate immune response all dealt with phagocytes. The adaptive immune response now deals with lymphocytes. Now, what exactly does lymphocyte means? Well, it's site, cell, and lymph means it's derived from the lymphatic system. So what exactly is the lymphatic system? Well, the lymphatic system is maybe another collection of systems that you may not be aware of. In HL, we looked at this system, and the major function of the system was actually collecting excess fluid from tissue fluids and returning it to circulation. But in the immune system, they have a super important role in these collection of lymph nodes scattered all around. So we're going to talk about the function of this in a second. Now, recall in cell specialization, we said that cells are able to differentiate from stem cells, but as adults we don't have very many, but we do have some stem cells in key areas called stem cell niches. One of these stem cell niches was actually located in the bone marrow, or the inside part of your bone. And the cells that we're concerned with here are something called hematopoietic stem cells, or HSCs. These are multipotent stem cells that are able to differentiate into any type of other blood cell. Specifically, we're going to talk about how do they differentiate into different lymphocytes. If we go through the differentiation process within a bone marrow, in here would be your HSC cells, and they're going to start to differentiate within lymphocytes. And then one of two paths can be taken. If these lymphocytes will mature in the bone marrow, then they would be ultimately forming B lymphocytes. If, however, they then migrate towards the thymus, which is an organ that's actually located behind your sternum, then if they mature in the thymus, they will ultimately differentiate into T lymphocytes. So differentiating in the bone marrow, B lymphocytes, differentiating in the thymus, T lymphocytes. Once they've matured in the thymus and the bone marrow, they don't stay there. Basically, once they've matured there, they're going to head off into the lymphatic system. And where they're mainly going to reside are in these lymph nodes and in the spleen. These are the two major reservoirs of your white blood cells. Now, something really interesting and really kind of brutal happens during the maturation process of B and T lymphocytes. And that is they're going to become hyper specific. Once we get a mature T or B lymphocyte, they are going to develop a specific receptor that is able to bind one and one only particular antigen. So this is a kind of a remarkable thing to consider. Within your body right now, you have over 2 trillion lymphocytes. And what's more amazing is of those 2 trillion lymphocytes, more than 100 billion of them are unique to each other, meaning that they're not genetically unique in that they're still going to have the same genome, but you can express over 100 billion different receptors, meaning that their receptors are going to have a binding site that is unique to each other. Now, how is that possible? This whole system is really remarkable. Now, this is outside the scope of the course, but it is really cool. But you have these enzymes within lymphocytes that are called RAG, or recombination activating gene proteins. And what they do is they actually bind onto certain parts of your DNA. And the sections that are actually going to code immunoglobulins, they can actually bind to it and recombine and shuffle those parts. So it's almost like splicing, but at a next level. And what this means is that they can form billions of different combinations of immunoglobulins. So immunoglobulins are made up of both heavy chains and light chains, and that top part here, called the variable region, that part is unique. So that part can only bind one particular antigen, and that's what makes your different T and B lymphocytes unique from each other. Now, a really important process happens within the maturation phase, and that is what's to stop your lymphocytes from creating a receptor that would bind one of your normal antigens that's found in your body. Wouldn't that mean that it's going to treat your body as foreign? Yeah, that would be a really big problem. So this is where the brutal nature of the maturation process happens. When they're maturing, something called negative selection is going to happen. So your body is actually going to expose self-antigens, or antigens that are found in your own body cells, and if any of those lymphocytes can bind to them, they're going to induce apoptosis and basically cell suicide and destroy them. In fact, this process is so selective that maybe about 1% of all lymphocytes will actually mature. Most of them are going to be destroyed to try to prevent any of them from incorrectly identifying yourself as being non-self. Now, it is worth noting that if any portion of these were to escape this process and still have these receptors, that's how you can get an autoimmune disorder, or a disorder where your own cells are attacking your tissues. This could be something that could occur in things like lupus or type 1 diabetes. In either case, your own body is attacking itself because it's incorrectly identifying self as non-self. Each one of your T cells are going to have a very specific T cell receptor. Due to that gene reshuffling, this shape is going to be very unique and is able to only bind one particular antigen. Your T cells also have another very important receptor, something called a CD4 co-receptor. And this co-receptor is going to help it bind to antigens that are going to be presented on your MHC class II complexes. So we'll talk about this role in a second when we discuss B cell activation. Now, your B cell receptors, these are going to actually, well, what does this look like? This looks like an immunoglobulin. Exactly. Their immunoglobulins are going to be receptors on their surface, and again, these regions are hypervariable and they can only bind one particular antigen. And that's what's going to lead to this specificity within the immune response. So to recap, your adaptive immune response all has to do with lymphocytes. Lymphocytes are originally derived in the bone marrow, and they can either mature in the bone marrow, B cells, or in the thymus, T cells. And during this maturation process, they're going to become hyper-specific, where they're going to express a receptor that can only bind one particular antigen that is a non-self antigen. In a process that we'll discuss in a second, your helper T lymphocytes are going to activate B lymphocytes. And the main function of B lymphocytes is to produce antibodies. So these are immunoglobulins. They also go by the name antibodies. So antibodies have a hugely important role in the adaptive immune response. Now, a quite large misconception about antibodies is that they destroy pathogens, but this is not true. They aid in the destruction of pathogens. So what exactly do they do? How exactly do they help destroy pathogens? Well, they can do this in a number of ways. The first way is through something called agglutination. And what this means, again, these antibodies can only bind to one particular pathogen on its antigen. So what agglutination is that a bunch of antibodies are going to basically lock together a bunch of these pathogens, and they basically clump them together. That's kind of another way that you can think about agglutination. So they basically attach in this kind of mesh-like structure a bunch of pathogens all together. This is going to greatly enhance their destruction in a number of ways. One, this is super going to reduce their movement. Imagine I was originally a pathogen and I can move all over the place really quickly, but now I'm locked together with all my neighbors. My movement is going to be much slower. So one, it reduces movement. Two, it makes it much easier for your phagocytes to actually engulf and destroy them. An analogy that I like to think about would be, imagine I have to clean the floor and I have to individually pick up little pieces of dirt. Or instead, I can take a broom and I can sweep it into a pile and then clean the pile. That's kind of how I think about this. It's much easier for phagocytes to destroy clumps of them than it would be individual ones of them. Another aspect in antibodies' aid in pathogen destruction would be neutralization. And what this means is that as a bunch of antibodies are bound on the surface of a pathogen, in this case a virus, it prevents the viruses from actually docking, because a lot of viruses and bacteria are able to infect by docking their antigens onto our cells' receptors. But if their antigens are all being occupied by antibodies, they have no way of physically docking anymore. So it prevents them from infecting or potentially destroying your own body cells. So this massively reduces their ability to infect our cells. And thirdly, opsonization is effectively marking cells for destruction. By having all of these antibodies locked on the surface of a pathogen, one, it helps your phagocytes identify an intruder. It's almost like spray painting a tree. Hey, cut this tree down. It marks it for destruction. But also it helps them bind to it easier for phagocytosis to occur. So overall, while antibodies don't directly destroy pathogens, they greatly aid in their destruction. And this is why they're so useful and a much more effective means of destruction than the innate immune response. Now, we've discussed the idea of antigens a lot, and let's formally talk about what they are. So really, an antigen can be deemed any molecule that triggers an immune response. And that kind of means that it doesn't necessarily need to be pathogenic. And a good example of this would be glycoproteins that we've explored in subtopic B1.1 as cell surface markers on, let's say, your erythrocytes. So recall that the human blood system is the ABO system. You can have four major blood groups: A, B, AB, and O, and they're all categorized by different glycoproteins on their surface. Now, let's say a donor has type AB blood. In this case, that means that they have the A and the B glycoproteins on their surface, and they want to donate that blood to someone with type A that only has A glycoproteins on their surface. If that's true, then what's going to occur in the body? So now let's zoom in to patient A's blood, and this is the donor's blood cell. It has both A and B glycoproteins. Well, the recipient's antibodies are not going to identify glycoprotein A as foreign, but they will identify glycoprotein B as foreign. This is a glycoprotein, this is an antigen that is not found in their own body. So even though the cell is not pathogenic, it is considered foreign, and this antigen is going to trigger an immune response. So a bunch of these antibodies are going to aggregate, and your phagocytes, the phagocytes of the recipient, are then going to destroy those cells. So even though the individual with type AB was trying to do a good thing and donate blood to this recipient, it would fail because this blood cell would be considered a foreign body or a foreign cell to the recipient's immune system. And in fact, this idea with a bunch of antibodies aggregating on the glycoproteins, agglutination, is actually how blood typing works. If I want to identify my blood group, what I would do is I take a little bit of my blood and I expose it to different solutions with different antibodies. A solution with antibodies A, a solution with antibodies B, a solution with AB, and a solution with O. And you basically are looking for where does agglutination happen. Now that we understand key differences between the innate and adaptive immune response, we're now going to look into how do the two work with each other to have a specific immune response. Let's finally talk about how the adaptive and innate immune response actually work together in order to provide immunity. Let's zoom in to the site of infection. So in this case, I've drawn little viruses, but keep in mind that this could be any pathogen or anything that's really considered foreign. So as we've already discussed, your phagocytes are going to initiate phagocytosis. Now, two different things are going to happen here. In some cases, your phagocytes are not only going to destroy them, but they're going to present these antigens on their major histocompatibility complex 2 or MHC2 receptors, thus becoming antigen-presenting cells. These cells are then going to go into the lymphatic system and then head off to your lymph nodes. At the same time, some of your phagocytes are actually going to destroy these pathogens and release these antigens through exocytosis. These antigens would also be collected by your lymphatic system and head towards lymph nodes. So at this stage, we have antigen-presenting cells presenting this antigen on their MHC2 receptors and free antigens entering the lymph nodes. We've now entered the lymph nodes. So what's happening here? So let's first talk about how your helper T lymphocytes become activated. So these antigen-presenting cells are arriving in the lymph nodes, and they have the antigens of the thing they just destroyed presented on their MHC2 class receptors. And they're really just trying to bind with any lymphocyte that matches. So this process is effectively random. It's going to continue to try to bind to a helper T lymphocyte that actually matches. Eventually, there'll be a match. So when the antigen on the MHC2 receptor is able to match complementarily to a T cell receptor and the co-receptor here, this complex will now cause the T lymphocyte to become activated. So again, this antigen presenting cell is effectively trying to bind to a number of lymphocytes. And what's occurring is that at some point, it could actually have a successful binding. Now, also in your lymph nodes are your B lymphocytes. Now, they too have not been activated yet, but recall that they have very specific antibodies on their surface. If one of those free antigens is able to successfully bind with that variable region on their immunoglobulin receptors, then receptor-mediated endocytosis will occur. And that just means receptor-mediated means it's being initiated or helped by a receptor, and we have endocytosis here. In fact, we looked at this process when we discussed clathrin. So antigens bound to antibodies are going to be taken in via endocytosis. Now, vesicles containing the same receptor that was found in phagocytes, MHC class 2, is going to bind and fuse with this vesicle, and now this antibody is going to move the antigen to this MHC2 class molecule. So similarly to an antigen presenting cell, this B cell is now going to present that same antigen on its surface. Finally, this activated T cell is going to ultimately be able to bind with this B cell, and this will occur in a few ways. Firstly, similarly to what occurred in the antigen presenting cell, the MHC2 class molecule with the antigen is going to be able to be sandwiched between the very specific T receptor and co-receptor. But in addition, the activated T cell is also going to release via exocytosis cytokines. These cytokines are going to be able to bind to the receptor on this B cell. So when you get binding between the MHC2 antigen and the T cell receptor, in addition to cytokines, then we can finally say that this B cell has become activated. Now, there's a point that I want to reiterate before I move on here, and that is both the B cell and the T cell did not develop specific receptors in response to an antigen. This is a very common source of confusion. Both the B cell and the T cell already had these receptors before this infection ever occurred. In fact, you likely have lymphocytes in your body right now with receptors to antigens that haven't existed yet. You should not think of this system as reactive in that it's developing this in response to antigens. It's that they already had so much variety that it just comes to finding the correct match. That's how you have to think about this system. It does not work by, oh, a new antigen, let me develop a specific T receptor, let me develop a specific antibody. No, both of these already occurred prior to infection. So what happens when the B cell finally becomes activated? They're going to start to initiate massive amounts of differentiation and division. So notice the difference between an inactivated and an activated B cell. This cell is what we would call a plasma cell. They have grown significantly in size, and they've developed significant amounts of rough endoplasmic reticulum and Golgi apparatus. Why would this type of cell need large amounts of these organelles? So recall that the main function of the B cell is to release a bunch of antibodies. What are antibodies? Proteins. These are proteins that are going to be released via exocytosis. Well, the function of the rough endoplasmic reticulum is to package proteins for exocytosis, and the Golgi is for modification. So both of these organelles are going to be crucial where these cells become an antibody-producing factory. In fact, these cells can produce upwards of 2,000 antibodies per second. That's how many are being released. So this cell, now that it's become activated and starting to divide, we now call this a plasma B cell. So this plasma B cell is going to start to undergo massive amounts of division. So it's going to undergo mitosis like crazy, making more and more and more copies. And this is something referred to as clonal selection, where it's only selectively making clones of the one B cell that was actually activated. That means that every single one of these plasma cells are making the specific antibody that can bind to that specific antigen that just entered the body. These plasma cells, like I said, are going to release thousands of antibodies a second, and that's all going to be released into the bloodstream. These antibodies are then going to be delivered via the bloodstream, ultimately to the site of infection. And these antibodies are then going to do everything that we just described earlier: agglutination, optimization, neutralization, and they're really going to turn the tide, so to speak, into the immune defense process. So now these phagocytes have this extra weapon of these antibodies that greatly facilitate their ability to destroy these pathogens. Not all of these activated B cells are going to become plasma cells. Some are going to become memory cells. Now, a memory cell is the same kind of B lymphocytes that produces the same specific antibody. However, unlike plasma cells that maybe remain for a few days to maybe weeks, memory B lymphocytes can remain in your body for decades. They're very, very, very long-lived. And this is what immunity is. This, I think, is a big source of confusion. A lot of people have the wrong idea about immunity. A lot of people have the incorrect notion that if I'm immune to a particular pathogen or a disease, it means that that pathogen can no longer enter my body. Of course, it could still enter your body through the same means that we already discussed. What immunity means is the retention of these B lymphocytes. By keeping these B lymphocytes in your body, if the same pathogen were to come back again, the response would be vastly accelerated. A good way of summarizing this whole process is with this graph. So here on the x-axis, we have the number of days from the beginning of infection onwards. And on the y-axis, notice that this is how many antibodies for a particular antigen you have. So the very first time that you encounter, let's say, a particular antigen, that will be day 0. And this would be during the primary immune response. So this is the first time that your body has encountered this antigen before. Everything that we just described would occur. So you would get phagocytes, antigen presenting cells, helper T activation, B activation, plasma cell generation, antibody production, and then hopefully the infection won't last too long, maybe a few weeks, in which case your body is able to overcome it and you would retain some memory B cells of this infection. Now, let's say at any amount of time in the future, you were to encounter the same antigen again. Well, if this person is immune, that means they've retained this memory cell. So if this immune person were to encounter the same antigen again, then these memory cells that have been retained are able to respond far faster, and they will immediately differentiate into plasma cells and produce a ton of these antibodies to immediately quell the infection. So notice that under the secondary immune response, the rate and the amount of antibodies produced is far higher than in the primary immune response. Look at how much faster antibodies are being produced, and look how much more antibodies are being produced. In fact, this response is so fast that this is kind of why you may not even realize you've been infected by something you're immune to. So really, when someone is immune to a particular pathogen, it means that their response to that particular antigen is so fast that really no adverse consequences typically occur, so much so that you may not even realize you've been infected by this thing. So this is really the power and the amazingness of the adaptive immune response. They retain memory of a particular past infection. And so this is why, while the innate immune response destroys anything, it doesn't really matter what it is as long as it's foreign, this is what you're born with. The adaptive immune response, as you can see, relies on experience. If you've experienced this infection, this antigen in the past, your ability to fight it in the future is vastly improved. It's almost as if you took a really hard test the first time, and that would be the primary immune response. And you're kind of struggling through this test. You may do okay, you may not do okay, but you kind of got through it. Everything's okay. Now let's say in the future, you wrote that same test again. Well, now you've already had exposure to that test before. You've remembered what all the questions were and what the answers were, and your result on the second time around is going to be much, much better than the first time around. That's kind of what the secondary immune response is. In my opinion, probably one of the most important scientific breakthroughs of all time is likely immunization. And this means that you are providing immunity to pathogens or diseases via vaccination. So unfortunately, vaccines have become divisive, but that is mainly to do with misinformation. Vaccines are probably one of the greatest piece of scientific technology that I can ever think about. Way back in the 1700s, Edward Jenner was actually the developer of the very first vaccines. Now, throughout human history, smallpox has been estimated to have killed well over 300 to 500 million people, and it was a large killer all around the world. And what Jenner realized was that these women that were milkmaids that were responsible for gathering milk from dairy cows, they were getting what was called cowpox, or a lesser version of that same disease. And what he noticed that these girls that were working with cows themselves did not get smallpox. So he made a hypothesis, and that was that their exposure to this lesser form provided some kind of protection against the more serious form. So what he did is he actually took some of the pus from the pustules from an infected milkmaid, and he took that and he injected it into a kid. And then that kid was then at a later time exposed to the real smallpox. And what ended up happening is that that child did not suffer really any consequences. He had some kind of protection because of the prior exposure to the lesser version. Now, he conducted this experiment much more times throughout other people, and he really provided the idea that exposure to this pathogen under less serious situations could still confer resistance to the full infection. And he coined the term vaccination, vaca from cow, because he literally used this in his original vaccine. So what exactly is a vaccine and how do vaccines work? So at a general level, how all vaccines work is they show or they provide your immune system the opportunity to experience a particular antigen, but without the risk of infection. So there's a lot of ways that this can happen. The most simple way that we can think about this would be what's called an antigen vaccination. And examples of this would be the HPV or the hepatitis B vaccine. And what's happening here is that we are injecting antigens into the individual. These antigens would then kickstart the same process that we just discussed. Phagocyte endocytosis, antigen presenting cell, activation of T and B lymphocytes, and then memory B lymphocyte production. So if the same individual was given an antigen vaccine of HPV, and then the real HPV virus infected that individual, they would already have retained memory B lymphocytes and were able to quell that infection very, very, very quickly. Now, another way that vaccines can work is that they may use inactivated or attenuated versions of that pathogen, or i.e. pathogens that are unable to actually infect the host. So these are non-functional versions of that pathogen. And examples of this would be the influenza or the flu vaccine, polio, MMR, and smallpox. All of these work in the same way, where we're inoculating or providing patients with the opportunity to see particular antigens, but again, without the risk of infection. And the same exact flow would occur. Phagocytes would undergo endocytosis to these inactivated or attenuated pathogens. They would then become antigen-presenting cells. They would activate T and B lymphocytes, and then create memory cells. Finally, and probably most impressively, is the mRNA vaccine. And this is a really, really cool system. So the mRNA vaccine is injecting mRNA or a code for the antigen in the first place. So in the case of the COVID-19 vaccine, we were injecting the mRNA code that is able to transcribe and translate into the actual surface proteins of SARS-CoV-2 or the spike protein. So what's happening is that this mRNA was then being transcribed and translated by your body cells that would then present the antigens on their surface. They would then activate antigen presenting cells, activation of T and B lymphocytes, and then memory cells. So again, they're providing your body the opportunity to see or experience these antigens without the risk of infection. To use my same analogy as before, if seeing an infection for the first time was like taking a test, kind of doing badly, and then learning from it, what a vaccine is, using that same analogy, would be you're taking the test for the first time, but it's a practice test. It has no bearing on your grade, it has no bearing on anything. So you can take it under much more relaxed situations, learn from it, memorize it, see what's going on, really understand it, and then at a later date, if you were to take the test again, you've already well acquainted with it, but there was no risk of you hurting your grade or failing or anything like that. And to use the same analogy, what would an mRNA vaccine be? Well, this would be like you have the digital version of the test, and you can just give the digital version to anyone. They can study it, they can memorize it. If they ever were to see this test again in the future, they're already well prepared for it. It cannot really be overstated just how important vaccination is and just how many lives this has saved. The direct consequence of vaccination has been an improvement in quality of life and in life expectancy. And the idea that we've basically hijacked our own adaptive immune response and used it in a way where we can develop an immunity against a disease without the risk of actually all the side effects that would come with a potential primary immunity. So overall, this system really, you can see, is an interplay between both the innate and the adaptive immune response, where your phagocytes are going to be the first responders. They then are going to activate T lymphocytes, which activate B lymphocytes, would differentiate into plasma cells and memory cells, and through the vast production of antibodies are able to quell an infection really, really quickly. This system, is my opinion, is just incredible. It's probably my favorite body system of all time. It is so, so cool, hence my bow tie. We've explored now both the primary and secondary immune response and the roles of the innate and adaptive immune system in fighting these infections. This is now going to be looking into a few case studies. And the first is going to be one of the most well-known viruses in the world, probably now only eclipsed by COVID because it's the most recent, but that would be HIV, or the human immunodeficiency virus. Now, the name HIV kind of already tells you what it does. Human, because it affects humans as its host. Immunodeficient means that your immune system is less so, or your immune system is compromised, or deficient, meaning it is impaired. And it's a virus. So this virus infects humans and compromises the immune system. It kind of already tells you what it is. Now, a big misconception between HIV is the difference between HIV and AIDS. Well, HIV is the virus, and someone may be HIV positive, meaning that they have the virus in their body, but they don't necessarily have AIDS. AIDS, or acquired immunodeficiency syndrome, is what may occur later in an HIV infection, which we'll talk about, but the two are not the same. Let's first discuss ways in which the virus can actually be transmitted from person to person, and then we'll explore how the virus actually infects your cells. There's a lot of misconceptions as to how HIV is spread from person to person. HIV is spread through the following bodily fluids: blood, semen, vaginal fluid, rectal fluid, and breast milk. Vectors where a HIV virus cannot be transmitted would be, a common misconception is through mosquito bites. So mosquitoes do not transmit HIV from person to person. HIV does not survive in saliva because saliva actually contains antiviral proteins, and so it starts to break down fairly rapidly. And of course, HIV is not transmitted by touching or shaking hands or sitting on a toilet seat, or there's a lot of misconceptions as to how HIV is spread. So let's explore the major routes of transmission. More than 75% of all transmitted HIV cases come from unprotected sex. And that sex can either be anal or vaginal sex. And that's again because HIV can be transmitted through the semen or through rectal fluids or vaginal fluids. So this is by far the most prevalent transmission process. The most effective way of stopping this type of transmission would be like stopping any other sexually transmitted infection, and that would be through the use of condoms. It's the cheapest and most effective way to actually block this type of transmission. Another means that HIV may be passed is through the sharing of hypodermic needles. And this is largely through the use of recreational drug use. So if I were to use, let's say, a needle to inject myself with heroin, and then I shared that needle with another person, some of my blood may still be in that needle, and it can transmit inadvertently to the other person. Another less common route, but still really worth noting, is that of blood transfusion. So if I'm donating blood, which by the way I encourage everyone to be able to do when they come of age, the people taking my blood are going to do a number of screenings to try to ensure that I don't have HIV, and they will likely test the blood before ever giving it to another patient. If you have HIV, you should not donate blood because that would be a very easy way of transmitting the virus from one person to another person. Another vector that people may not be aware of would be that HIV can actually be transmitted from an infected mother to her fetus or to her newborn child. And that's because HIV can actually pass the placental barrier, or it could actually be transmitted through breast milk. So this is something to really keep in mind, that if the mother is HIV positive, she could be transmitting it to her fetus or to her newborn child. What kind of host cells does HIV infect? HIV can't infect all of your cells, but cells that are very, very specific, or something called CD4 positive cells. And what this means is any cells that have the CD4 co-receptor on their cell membrane. So the ones that we've explored in this course would be helper T lymphocytes. These are going to be the primary cells that we're going to focus on. So recall that helper T lymphocytes, in addition to having a specific T cell receptor, they also had a CD4 receptor. And so this is what the HIV virus is actually able to dock to. So HIV have glycoproteins on its surface like any other virus, and this allows them to actually bind to the CD4 receptor on the helper T lymphocytes. Once it does this, HIV can then fuse with the lymphocyte membrane. HL students, we've explored in the virus subtopic that an HIV virus, in addition to having a protein capsid surrounding it, like all viruses do, here shown in gray, they're also what's called an enveloped virus, meaning that they have a plasma membrane surrounding their shell as well. And this is what allows them to fuse with the host cell and then allow them to inject their contents. In this case, their contents are going to be RNA because they're an RNA virus, and they're going to also inject some key enzymes in here that we'll explore what they do. So HIV is actually a retrovirus, meaning that it has an enzyme that is able to convert its RNA into DNA. Now, in our process, we were able to convert DNA to RNA in a process called transcription. So they have an enzyme called reverse transcriptase, which, as you may imagine, does the reverse process. It is actually able to convert their single-stranded viral DNA into double-stranded DNA. After that, they have another enzyme that they come packaged with called integrase. And again, this name is actually quite intuitive. It now allows this double-stranded viral DNA to integrate within the host's DNA. And now the helper T lymphocytes DNA, a portion of it, has this now new section that contains genes that are now going to be able to code for more viral proteins. Now the host cell, without really realizing, is when it undergoes transcription, it is going to be able to transcribe these viral genes in order to make more viral proteins. These viral proteins are then going to be able to assemble into brand new HIV viruses. And then they're going to fuse with the host membrane, become enveloped again, and then allow them to infect a brand new helper T lymphocyte cell. So in effect, the helper T lymphocytes have effectively become machines of making more and more and more copies of the HIV virus. Now, understanding the progression of an HIV infection can be kind of understood well using a graph that looks like this. And you can follow along with my notes that are found to the left of this. But really, we can break down an HIV infection into three major phases, here shown as A, B, and C. Now, let's explore what this graph is showing. On the x-axis is the time from infection all the way through its progression, and on the y-axis, in blue, is the helper T lymphocyte count, or how many cells per volume. Usually this is expressed in number of cells per millimeter cubed, or per milliliter. And in red is how many copies of the HIV virus is actually present in the individual. So in phase 1, we have the acute phase, and this is where the initial HIV infection would occur. And so the moment that HIV is able to successfully enter a host, they're going to replicate very, very, very quickly. As you see in this very sharp rise in HIV copies, they're going to find all kinds of free available helper T lymphocyte cells, and they're going to start infecting very, very quick. As you may imagine, the T cell count is going to mirror this, whereas HIV copies go up, the T cell count is going to go down. And the reason for this is as helper T lymphocytes actually start making more and more and more copies of HIV is going to burst out and destroy or kill these cells. So this is going to cause death of helper T lymphocytes. Now, your body is going to be able to mount a response to this like any other infection, and this is going to quell the numbers, so to speak, but it won't completely eliminate them. A lot of the HIV is going to be able to remain dormant. by still having part of their viral DNA within host T lymphocytes, and they're going to be able to enter what's called the now phase 2 or latent phase. And in this phase, they are still infecting helper T lymphocytes, but a significantly reduced rate and degree. This phase may last a few years to potentially over a decade. So this is where the break here in my timeline, because this could last a very, very long time. But what you are going to notice is a slow increase in the number of HIV particles and a slow decrease in helper T lymphocyte quantities. So you're going to see this, again, this mirror image curve, where as HIV starts to go up, helper T lymphocyte cell count starts to go down. So when is someone actually going to be diagnosed with AIDS? What does that mean? AIDS is defined really by a specific amount of helper T lymphocytes in your blood. The count is typically less than 200 helper T lymphocytes per milliliter. So that's typically when someone is going to be diagnosed as AIDS. So on this graph, that would be less than 200 here. You don't have to know that number specifically, but this is now when someone would actually be diagnosed with having AIDS. Someone who has AIDS now is at really high risk. Well, why does having a low helper T lymphocyte count really matter? Well, as we've already discussed, the helper T lymphocytes are a crucial aspect of your adaptive immune response. And what happens is that a lot of people that actually end up suffering from AIDS and potentially die as a result don't die directly from AIDS, but what are called AIDS-related infections. So a common one would be like pneumonia, where pneumonia being a bacterial infection of the lungs, and usually someone would be able to fight off this type of infection. But because their immune system is so compromised, they are usually unable to fight off this infection. And a lot of people that end up dying as a result of AIDS die from these other AIDS-related illnesses. So how exactly do we treat someone with HIV and AIDS? Is there a cure? Unfortunately, no. There is no cure for AIDS. That may seem a little odd. Can't we develop a vaccine like we have for polio, like we had for COVID-19? Why is it that we don't have a cure for AIDS yet? Surely this has been occurring since the 80s. What's going on? Well, during the process of converting single-stranded viral RNA into double-stranded DNA, reverse transcriptase, unlike other enzymes, lacks any proofreading capabilities. That means that it actually makes a number of mistakes or mutations fairly often. That means that there are potentially many different versions of HIV being made in the same individual. And so again, to your immune system, they recognize pathogens by these glycoproteins. And if you change the shape of these glycoproteins to your immune system, that's kind of like a brand new thing. So when you're developing a vaccine, we're developing vaccines in response to specific antigens. And if these antigens continually change, you have to continually craft new and new vaccines and treatments for this. And this is kind of how during the COVID epidemic, if you recall that there was COVID Delta and Omicron, what this was it's the same virus, but slightly different variants because the spike protein was changing in shape. The same thing is happening with HIV. Because reverse transcriptase makes a lot of mistakes or mutations, there's a lot of different variants of HIV, and that makes it very, very difficult to cure. There's been a lot of improvements over many years in being able to treat HIV, so much so that someone that is diagnosed with HIV and potentially AIDS, their prognosis has significantly improved over time due to these different kinds of treatments. So if you really consider how HIV works in terms of an infection, you can try to target different stages of that infection, and that's how these treatments work. So for instance, there are CD4 inhibitors. So this would actually block HIV from docking to your T lymphocytes in the first place. So this would reduce the amount of newly infected cells. You could also inhibit reverse transcriptase. This would prevent an HIV from actually replicating within a host cell. Or similarly, you could also inhibit integrase, and this would prevent a double-stranded viral DNA from integrating into the host DNA. So once you understand the mechanism as to how a pathogen infects, you can then target various elements of that infection process. And that's how these drugs work. And again, as mentioned, because of the very high rates of HIV mutation, It is possible that HIV can become resistant to any one of these treatments, so it's actually quite common for a combination of these different medications to be used. Now, something that's often conflated or confused is the difference between viruses and bacteria. A lot of people just treat both as germs, but they are very different types of pathogens and they must be treated very differently. So unlike viruses, bacterial infections can be treated with antibiotics. An antibiotic is a specific chemical that specifically targets prokaryotic cells. Let's explore how they work. Similarly to what we just discussed with treatments for HIV, once you understand the metabolism and how a specific organism actually replicates and works, you can target various aspects of that process. So in terms of antibiotics, they're going to target processes that are specific to bacteria. There are many different classes of antibiotics, each of which target a slightly different part of the bacterium's life cycle. Now, this was partially explored in the enzyme subtopic at the HL level, where we actually explored the function of penicillin. Now, penicillin as an antibiotic is a type of an antibiotic that actually blocks or targets an enzyme within bacteria that builds their cell walls. Now, their cell walls are made up of peptidoglycan. So how this antibiotic works is it actually inhibits that enzyme, and as a result, the bacteria cannot build strong peptidoglycan cell walls. Well, what does that mean? Well, due to the turgor pressure within their cells, they no longer are able to resist it due to having weakened peptidoglycan cell walls, and thus they're actually going to lyse, or they're going to burst open and thus killing these cells. So these types of antibiotics are able to break open or really destroy bacteria using this mechanism. Another one, similarly, is that it will target cell membrane permeability. So it will make the cell membrane much more permeable, and again, it could lead to cell lysis and death. Another very effective means of antibiotics is actually to target the protein synthesis process. If you block an organism's ability to do protein synthesis, that organism will die. So there are various ways that you can actually do this. For instance, you can stop it at the transcription level, or you can even stop it at the translation level. There are some antibiotics that actually can inhibit RNA polymerase, or they can inhibit a new tRNA from actually binding into a ribosome, or they may even inhibit peptide bond synthesis during the translation process. So if you inhibit transcription or translation, these bacteria can no longer produce proteins that they need to survive. One of the aspects that makes bacterial infection so taxing on the body is that they're able to replicate very, very quickly. So some antibiotics actually target this aspect of their metabolism. So they can actually target certain enzymes that are required for DNA replication, such as topoisomerase activity. This enzyme actually causes the DNA chromosome to unsupercoil and thus allows DNA synthesis to occur. So if this antibiotic prevented this enzyme from functioning, this means that DNA synthesis is no longer possible. So if you can't replicate your DNA, you can't replicate the cell, bacteria can no longer replicate. So how all of these antibiotics work is that they either directly destroy a bacteria by weakening either their cell walls or cell membranes, or they can impact a crucial aspect of their metabolism, such as transcription, translation, or DNA synthesis. Now, it is a really important thing to note that in all of these mechanisms of action, they only target structures and enzymes that are found in prokaryotes, not found in eukaryotes or found in viruses. If I take an antibiotic, why would it not target my own cells? Well, my cells lack peptidoglycan. My cell membranes have a different composition than that of prokaryotes. While yes, my cells will undergo transcription, translation, and DNA synthesis, they don't use the same enzymes. So these enzymes that are used in prokaryotes are not the same enzymes used in eukaryotes. So all of the targets that antibiotics have are specific to prokaryotes. Now, this aspect is not just important for understanding why antibiotics don't harm you, but why they won't have any effect on a viral infection. The flu or the common cold, these are viral infections. And what ends up happening is people use the idea of antibiotics as, oh, if I'm sick, I'll take an antibiotic. But actually taking an antibiotic during a viral infection can have really negative repercussions. And a reason for that is that not only will it not destroy a virus, it will instead start to destroy other bacteria. And your body is home to trillions of actually beneficial bacteria, such as in your gut. So this could actually harm bacteria that are actually helping you in your body and have no impact whatsoever on the virus that is currently wreaking havoc in your system. So antibiotics should really only be used in fairly serious bacterial infections. So if antibiotics are so good at killing prokaryotes and pathogenic bacteria, why are they still a thing? Why are they still such a problem? Well, that's because, like all living things, bacteria can evolve. And what we're seeing, and what we've been seeing over a long time, is bacterial evolution in the way of resistance to antibiotics. So let's explain how this process works. Now, the process of evolution via natural selection will be explored in subtopic D4.1 when we discuss natural selection, but I'm going to explain how this process can happen in the context of bacteria and how they can develop antibiotic resistance. So a very big misconception that I want to get out of the way before I get into evolution is that what bacteria are not doing is experiencing an antibiotic and then in response developing resistance to it. That's not what's going on. Bacteria are not evolving an adaptation in response to an antibiotic that they've experienced. How natural selection works is it all has to do with mutations. A mutation provides variation. If that variation enables a competitive advantage, then that increases the likelihood that that individual will reproduce, pass on that advantage to their offspring, and then over time, that population will have that advantageous characteristic more and more and more. Thus, we would never say that an individual is evolving, but we would say that a population is evolving. An individual does not adapt, but a population may become adaptive. So let's describe how this happens in the context of bacteria. So to begin with, why is it that we're seeing evolution so quickly in bacteria, whereas let's say in humans, we're not seeing such radical changes? And really, it all has to do with the reproductive rate. We undergo mutations all the time, just like bacteria do, but our reproduction rate is very, very low, and our lifespan is very long. So that means it would take many, many years to see multiple generations of humans, whereas in bacteria, for instance, in the case of E. coli, you can get a brand new organism in as little as 30 minutes. So bacterial populations can explode exponentially in a very short period of time. So because there are so many opportunities for reproduction, it means that the likelihood that mutations can develop and be spread out is much, much higher. So this is why we see so many different mutations over time in bacteria. Even though maybe their mutation rate is actually not that high, there's just so many of them that it becomes a numbers game. Now, let's imagine that a random mutation happens, and recall that a mutation is just a change in the base sequence of DNA. And let's imagine that this mutation is actually going to confer some degree of resistance to an antibiotic. Now, what could that actually look like? How does a mutation cause a bacterium to be resistant to an antibiotic? Well, there's a number of ways that this can happen. These are all examples of mutations that we've already experienced. So what are some examples of some mutations that have conferred resistance? There could be a mutation that actually prevents or reduces an antibiotic's ability to even enter the cells. So an antibiotic may still be present around this individual, but this mutated individual cannot actually get the antibiotic entering them, thus rendering it ineffective. So in another scenario, there could actually be a mutation that leads to a new enzyme, and this enzyme is able to actually degrade or break down this antibiotic before it's able to actually work. There could be a mutation for a particular type of protein that actually pumps out the antibiotic. So the antibiotic in this case would still be able to enter this mutated individual, but they're producing a protein pump that basically just sends that antibiotic right back out, again rendering it ineffective. And lastly, another cool example of an antibiotic resistance would be that the target is actually modified. So the antibiotic would typically be able to, let's say, target a particular enzyme, but a mutation actually slightly alters the shape, disallowing this antibiotic from actually binding and being effective. And these are just some examples that we've already seen in antibiotic bacteria. So in a population of bacteria, due to random mutation, one of these individuals potentially has one of these adaptations, and they have this mutation that allows them to be resistant to, let's say, antibiotic A. And so in the presence of antibiotic A, this one individual now has a significant increased chance of survival. So what will this look like in terms of evolution? Well, if that bacterium that now has this mutation is in a host that is now taking this antibiotic, let's call it antibiotic A, what happens? That antibiotic is now acting as a selection pressure. It is now going to effectively select for any individuals that are resistant versus non-resistant, because in the presence of this antibiotic, most of the population of bacteria are going to die or are going to perish. Because antibiotics, as we've already described, are very effective at destroying bacteria, or at the very least weakening them to such a degree that the host's own immune system could destroy the bacteria. But in the case of this mutated individual, that antibiotic is not effective against them. So what that means is that while most of the population has potentially been killed by the antibiotic, these individuals with resistance survived. And those individuals will then reproduce via binary fission. And recall that binary fission is making clones of an individual. So that means all of the offspring of that mutated individual with antibiotic resistance also have antibiotic resistance. So in effect, this new population of bacterium over time will all be antibiotic resistant. So in the future, if that bacterium's host were to take antibiotic A again, they would not be affected. So they have developed as a population antibiotic resistance. Note that in this theoretical example, the bacterium did not develop this mutation in response to the antibiotic. They already had that mutation. And in the presence of the antibiotic, it was suddenly adaptive. So really a key thing to understand about evolution is something is only adaptive in a correct scenario. Something that was adaptive in one environment may actually be disadvantageous in another. So it's really context dependent. So not only can bacteria transfer adaptations down a line of inheritance, as in from parent to offspring, but they can also do what's called horizontal gene transfer. And something that we looked at when we looked at the structure of cells, some bacteria have pili that enable them to actually link with adjacent bacterium in a population. And what they can do is they can actually transfer a plasmid or a small section of their DNA and transfer it to another member. Now those plasmids typically hold onto genes that are adaptive, or in the case here, confer antibiotic resistance. So this means that these individuals that, let's say, have this antibiotic resistance can encounter another bacterium that does not have antibiotic resistance, and they can literally transfer those genes to that individual. So it's almost like I have a USB with a particular file and I'm giving it to somebody else to share. This is not reproduction, but they're still able to share genes in a population. So this allows population of bacteria to become resistant fairly quickly. This is what allows bacteria to actually be fairly adaptive in any given environment. They replicate really quickly. This allows multiple mutations to potentially arise in a population, and due to both vertical and horizontal gene transfer, it allows these potentially adaptive characteristics to be transferred fairly quickly throughout a population. Now, this wouldn't be so bad, but we as humans have exacerbated this problem. How have we gotten to a point where we now have bacteria that are resistant to nearly all types of antibiotics? These are sometimes referred to as superbugs. Probably the most famous example here is MRSA, or methicillin-resistant Streptococcus aureus, or what's on my bow tie right here. This is the classic example of a superbug or a antibiotic-resistant bacterium, because this species is resistant to multiple different kinds of antibiotics. But how did this superbug actually develop? The problem is that people are overtaking antibiotics. And so what I mean by that is, for one, let's say I get sick, I get a cold, I have a fever, I feel sick. So the first thing I'm going to do is I'm going to take antibiotics because that's going to fix the problem. But what if what was causing me to be sick was actually viral? If it was a cold, that's viral. If it's a flu, that's viral. If it was COVID, that's viral. So taking an antibiotic is actually entirely ineffective. What I'm doing is I'm giving populations of bacteria, potentially in my body, a selection pressure to potentially have this resistance be able to be propagated more. What are some steps that we can do to try to mitigate this issue? One of the major reasons why this has become such a problem worldwide is that it's not just humans that are taking antibiotics, we also use antibiotics in livestock. And so imagine we're holding a bunch of chickens, a bunch of cows, a bunch of sheep in a very close proximity. That also allows a higher degree of pathogen transmittability. And we don't want our livestock dying, so we give them antibiotics. But the problem is that by giving them more and more and more, these animals, just like us, are given the same opportunities for their bacterium to actually spread antibiotic resistance, and then that can then spread to humans. So the idea is that it's not just human overtaking antibiotics, it's also livestock overtaking antibiotics. And so the first thing that we should do is we should reduce the use of some certain kinds of antibiotics in farm animals to try to mitigate this problem. Secondly, we would want to restrict the use of antibiotics to only really serious bacterial infections. We don't want to use it just whenever someone is sick. We want to really reserve it for really cases where antibiotics need to be used. Thirdly, if you are prescribed an antibiotic and there's a recommended dosage and a recommended length of time to take it, please stick to that recommendation. What ends up happening is someone will take some antibiotics, they'll feel good maybe after a couple of days, but they still have another week left of their course and then they'll stop. Well, again, this may allow antibiotic resistance to actually continue to propagate. So you really want to make sure, regardless of how you're feeling, to complete the course of antibiotics to try to mitigate this problem from happening. Like many other nature of science learning outcomes, this is referring to scientific techniques and how new techniques can lead to new avenues of research. And in this case, what does this have to do with antibiotics? Well, we have chemical libraries now where we've been able to sequence and determine the shape and structure of hundreds of thousands of different proteins, and this allows potentially new different antibiotics to be used as chemicals or even different targets that could be assessed. Now, we are pretty good at doing this, but a big advancement that's really going to help this is through the use of AI and deep learning with respect to searching these libraries and coming up with new potential targets. So AlphaFold is an AI program that has been trained on thousands of different protein folding configurations, and it is now able to fairly accurately predict the structure of a protein given a gene sequence or given an amino acid sequence. There's been a lot of technical advancements that can really help us turn the tide, so to speak, in the fight against antibiotic resistance. One of those could be the ability to better diagnose antibiotic-resistant bacteria in an individual. Currently, we can do it, but the wait times could actually be fairly long. And so how can we actually improve this technique? Well, AI has been able to be used to actually potentially determine single-cell susceptibility given a specific sample, and this can be done within hours. And this technique is becoming much more prevalent and more understood. AI and AlphaFold can potentially be used to not only comb different huge databases of chemical structures, but it also potentially identify new chemicals that could be used as antibiotics and new targets for those antibiotics that are found in bacteria. And so using different testing, we can try to identify and test, would this chemical be able to now do this to this bacterium, and so on. So we can even use gene knockout, where we can actually try to identify which proteins, which chemicals can have what effects on bacteria, and this will allow the development of new antibiotics in this fight. So most pathogens are really specific in terms of which host they can actually infect, meaning they are species specific. So for example, the pathogens that cause polio or measles in humans would not be able to cause those same diseases in other hosts. They would be ineffective. So why is that? Why is it that pathogens tend to be really specialized and specific is to as to what host they can actually infect. Pathogens may require a specific receptor, meaning that in order for this particular pathogen to infect a cell, they have to be able to dock or bind to, let's say, a protein or a glycoprotein that's only found in this particular species. So in that sense, they're very specific as to what host they can actually infect. So pathogens may be able to enter hosts, but they won't be successful in terms of their metabolism, meaning that their growth rate or their metabolic rate is going to be much below optimum because maybe, let's say, the body temperature of the host is different than what their optimum temperature is, or perhaps the pH or the salinity of the host is different than that of their own. So they have to have compatible physiological conditions that allow their metabolism to be effective. And lastly, pathogens may require a particular nutrient or substrate. So they may be able to enter the host, they may be able to dock or infect cells, but in order to grow, they need a particular nutrient or particular substrate in their metabolic processes, and the host lacks this. So they're going to be ineffectual at actually, again, growing and multiplying and not be able to be successful. So for all these reasons and more, typically pathogens are really species specific. However, unfortunately, there are cases when pathogens can what's called cross the species barrier, meaning that they used to be specific to this particular host and they have jumped to a different host. Zoonosis is a pathogen or a disease used to be contained within a particular organism or maybe a few particular organisms, and for whatever reason, they are now able to pass that on to humans. And this can lead to really rapid infection rates and really rapid spread because this is the first time that we've encountered it. This is the first time that it's impacted humans. So for this reason, this can lead potentially to an epidemic or a pandemic. And these words should not be unfamiliar to the people watching this because we just went through both of these very recently with respect to COVID-19. So what exactly is the difference between an epidemic and a pandemic? It really has to do with the scope of the spread of disease. So an epidemic typically is an increase in disease occurrence above what would be expected in a particular area or a particular population. So as a city, as a town, as an area, they are typically, we would use the terms epidemic for these reasons. So when do we use the terms like pandemic? Well, a pandemic is really over a much broader area. So this could be over a whole country, multiple countries, multiple continents. So when the WHO announced that COVID-19 was we were officially going through a pandemic, that term would not be used lightly. That means that multiple areas over large geographic scope were experiencing higher rates of infection, much above what we would expect. But let's look at a few examples of zoonotic diseases. And keep in mind that there are many, but the course lists 4 key examples that we're going to look at. So let's begin with arguably the biggest killer in human history, that being tuberculosis. So this is a very old pathogen. This has been around for a very long time. Even if we look at human history, we found evidence of mummies having tuberculosis. So that stretches back 4,000 plus years. So the pathogen, as a zoonotic pathogen, is Mycobacterium bovis. Bovis meaning that it used to be reserved to cattle. Cattle used to experience tuberculosis. And then at some point, this bacterium was now able to be transferred to humans. So there are 2 major clear vectors or 2 major routes that this particular bacterium can be transferred from a cow to a human. That being 1, drinking unpasteurized milk from an infected cow. So if I were to milk this cow and then immediately drink this milk, that can absolutely contain this bacterium and I can be infected as a result. Today, the common method of drinking milk is to first pasteurize it. This is named after Louis Pasteur, but pasteurization really just means to heat the milk in order to kill any pathogens present in the milk prior to actually consumption. So this absolutely should be done. Consuming raw milk is incredibly ill-advised for this reason and more. The other mechanism of infection could be, well, cows are mammals and cows cough. So if the bacterium is currently infecting the cow could cough, bring up droplets, and then those droplets can then be inhaled by a person nearby and they can be infected. So this is a great example of a zoonotic disease because it used to be in cattle and it can be transferred to humans. Now it is worth noting that there is another strain that infects people specifically, but either way, this disease is a very, very prevalent one. John Green just wrote a book about everything is tuberculosis that effectively is almost like a overview as to how tuberculosis has kind of been a big part in shaping human history. It's a really fascinating read. What exactly does tuberculosis do? Why is it such a problem? Well, it infects the lungs. The bacterium will be able to be inhaled. They will then start to grow and spread within the lungs. And then what are the classic symptoms of someone with TB or tuberculosis? And that is coughing blood. How is that happening? Well, it causes lung damage. The alveoli walls could be breaking. They could be causing serious tissue damage. And then as you're coughing, what you're expelling is basically broken or dead tissue from the lungs. So there's also, what are the classic symptoms is very pale skin. This goes by another name of consumption. We have treatments and we have vaccines for tuberculosis. However, these are not worldwide spread, and depending on the access of medical care that some countries and some people have, they can still absolutely be infected and can potentially die from tuberculosis. So to this day, there is still over 1 million deaths per year due to tuberculosis. So this is a really big problem. Another fairly well-known disease is that of rabies. Now, unlike tuberculosis, this is caused by a virus. It's caused by the Lyssa virus. And this actually comes from the Greek word for the Greek god of rage and fury. And that's because rabies really induces a really visible change in behavior of the person that is affected. It's probably widely believed that this virus is the inspiration for zombie outbreaks. Because how is it transferred? It's transferred by biting, and it results in really erratic behavior in the host. So let's talk about this a little bit. So the Lyssa virus infects many different vertebrate mammals, such as dogs, bats, raccoons, squirrels. So it does spread fairly wide in terms of the mammal world. The most common cases of human infection, though, come from dogs, and that's likely because our close regular proximity to dogs. So if a dog becomes infected with rabies and they bite humans, meaning they're breaking skin, the virus is actually in the saliva, and so that can be transferred directly into our blood. Another way could be from a scratch from an infected animal. So the infected animal, it can scratch you, and then the virus may be able to enter through the open wound. What does rabies do? Well, rabies has a really long, what's called incubation time, meaning that from the time at which you are being bit to the time of presenting symptoms is actually quite long, meaning that I could be bit by a dog that has rabies and I won't experience symptoms potentially for many months later. However, what's quite scary about rabies is that if I do start presenting symptoms, this virus has a 100% mortality rate, meaning that once symptoms start presenting, there's really nothing that physicians can do to help that person, which is quite scary. So why is it that millions of people aren't dying from rabies every year? Well, because once I'm bitten, because the incubation time is so long, it means that I can get treatment in that time. And if I get an anti-rabies treatment or a vaccine, this should easily be able to curb and end that infection, and I will be fine. Rabies is actually, once from the point of being bit, it will actually travel through your bloodstream and then make its way to your nervous system, or your central nervous system, and into the brain. And it will start causing some big problems in the brain. And one of the major reasons why someone can die from this is inflammation, meaning the brain is going to start to swell. And as the brain swells inside of a immovable cranium, you can start causing tissue damage. Because we have regular treatment in most of the world, the yearly cases of death are not that high. However, it is still resulting in nearly 60,000 deaths a year. And that's mainly due to, again, people that don't have access to treatment and are not exposed to medical care as widely as, let's say, someone would be where I live in Japan. Another zoonotic disease that you may not have heard of is something called Japanese encephalitis. This obviously gets its name from it was originally discovered in Japan, but this is not just present in Japan. It's present mainly in Southeast Asia. And so this is caused by, easy to remember, the Japanese encephalitis virus. And this virus actually has probably the most complex life cycle of all the ones that we've looked at before. And so the virus is going to be present in large numbers. The viral load is going to be quite high in animals like pigs and wading birds. And this is where they're going to be replicating, and this is where their numbers are going to be really high. And the way in which this continues to be high is that mosquitoes can bite an infected pig, transfer it to another pig, transfer it to another bird. And in this way, this is what's called the viral reservoir. This is where most of the viruses are going to be, is they're going to be transferred from pig to pig or pig to bird through mosquitoes as the main vector. If a mosquito bites an infected pig and then bites a human, then this is what's called an end or a terminal host, meaning that they will now infect a human, and the human will not be able to infect other people. And that's because our viral load is going to be actually quite low. So the cycle is that mosquitoes bite infected pigs, transfer it amongst other pigs and other birds. But if that mosquito were to bite an infected pig and then us, we are now infected by Japanese encephalitis. What does the name encephalitis mean? Encephalitis mean? Well, encephalon means brain, and itis usually means an inflammation. So it literally means an inflammation of the brain. And so once a human is infected with Japanese encephalitis, what could happen is their brain again can start to swell. And as we've discussed, as a brain starts to swell inside a cranium, then the blood vessels, the tissue of the brain can start pushing up against the skull. And that can cause tissue damage, that can cause major brain injury, and it can even cause death. So this still causes upwards of 20,000 deaths per year, mainly in Southeast Asia. And the people that are mainly affected are those living in poorer rural areas, again, with lack of medical care, and where they may not even be aware of this disease or how it's actually being transferred. And then finally, a virus that I don't think requires any introduction, and that being the SARS-CoV-2 virus. So we've explored this virus in HL, in the virus subtopic, but for the record, the name of this virus is not COVID-19. COVID-19 is the disease. The name of the virus is SARS-CoV-2, or Severe Acute Respiratory Syndrome Coronavirus 2. So again, coronavirus comes from the fact that it has corona, or a crown of glycoproteins surrounding it. And coronaviruses are almost like a group of viruses. There are many different kinds of coronaviruses. So why am I talking about this with respect to zoonotic diseases? Well, it is hypothesized that this is a zoonotic disease, and it transferred very, very recently from an animal to humans, and the hypothesized animal is that of bats. So this is the, at least, hypothesis now as to, and this is why cases rose so quickly in Wuhan and then quickly throughout the world, because infection is very easy. And again, symptoms do not present themselves very, very early. So people can be infected and maybe not know it yet. So the coronavirus will be able to enter your lungs, and they're going to start a number of respiratory symptoms. And as you well know, you can survive COVID-19 and recover or there are many, many deaths. As of today, there was well over 7 million dead around the world from COVID-19. A big reason why we think that the cases spread so quickly and so fast was because it was a recent zoonotic disease and we just weren't prepared for it. Okay, so we just talked about all the ways that humans can start getting infected and spreading it around the world. Well, what can we really do about it? And to really understand this is the concept of herd immunity. So what herd immunity is, it's trying to resist the spread of a disease in a population by ensuring that the majority of the population is immune to that disease. And this could happen, as we've already discussed. Recall that being immune means that the individual has retained memory cells of a particular pathogen. So becoming immune can happen in one of two ways, either from being infected, recovering, and then keeping memory cells, or it could be developed through vaccination programs. So let's discuss a little bit about how this actually works. The concept of herd immunity, I think, is fairly intuitive, and that is the more people that are immune to the pathogen, the less likely that pathogen can actually spread, because if it spreads to somebody who is immune, it cannot successfully incubate there, and it will be killed off very quickly by that person's immune system. The greater and greater the portion of the population that becomes immune becomes, the less and less the rate of spread will be in that population. Now, the best way to do this is through vaccination programs. So the way that this works is that the members of the population are effectively interdependent, meaning that they rely on each other. I think really the core of herd immunity and vaccination program is really collectivism, and that is for the group. And that is because I'm not just vaccinating myself to protect myself, I'm also doing it to protect others, especially those vulnerable members of the population. There are people in the population that are going to be unable to receive a vaccine, and that could be because they have an allergic reaction to certain chemicals, certain reagents within the vaccine, or the vaccine is usually restricted to certain age groups. And if you're too young, especially, you are unable to get this vaccine. Now again, these are the most vulnerable members of the population, those people that are maybe immunocompromised, those people that are very, very young, they can't get vaccines, so what do? Well, if we shield them, so to speak, by having the majority of the population be immune, then the likelihood of them contracting this illness becomes very, very low. So let's take a look at how this works. Let's imagine an initial scenario here where there are no immune members in this population and two different members have become sick. And let's imagine that this illness is spread through coughing, like a lot of illnesses are. This means that the amount of people that they can each infect becomes fairly high, and then over time, this can spread exponentially through the population. And because no members of the population are currently immune, the case numbers can rise very quickly. So this is what effectively happens at the very beginning of an epidemic or a pandemic, where nobody has experienced this pathogen before and case numbers start to rise very, very quickly. If the majority of the population now becomes immune, and recall that there's still going to be some people that maybe can't get it, yes, you can still get this pathogen, this disease to spread, but the amount of people that get infected is going to be significantly reduced because there are other members of the population that are immune. So this means that these people who are still contagious, if they're right now coughing, these members can still get the pathogen, but because they're immune, their immune system is fighting this so quickly that it is of high likelihood that that pathogen is going to be destroyed very, very quickly, and the likelihood of them being able to transmit to somebody else is also fairly low. So the idea is that more and more people that are immune, the lower and lower and lower the rate becomes. Now, it should still be noted that even though in, let's say, a case of an epidemic or a pandemic where we're really trying to curb the spread, even though I am immune, it's still recommended that you reduce any chances of spreading it. So in the case of COVID, which was transmitted through coughing, it was still advised to wear a mask despite the fact that, let's say, I would have had immunity, because there is still that minor chance that if I'm infected with COVID, I can spread it to somebody else. So wearing a mask is still very, very highly preferred in order to curb any spread. And this has been shown in countless of different studies. How do we determine how much of the population, what percentage of the population should actually be immune in order to have herd immunity? And that's where this nifty little formula comes in. And this relies on something called the R0 value. So the R0 value is effectively the mean number of people that an infected person will infect. So if there is an R0 value of 2, then this means that if you're infected with this pathogen, then on average, you're going to infect 2 more people. So if this is the case, if the R0 value was 2, then what percentage of the population should be immune in order to have successful herd immunity? So I can do 1 minus bracket 1 divided by 2, close bracket multiplied by 100. If 50% of the population were immune, then the population as a whole would be able to develop herd immunity, and the likelihood of spread gets significantly reduced. Now, it is not necessary for you to know this formula, but I do think that it is quite handy and quite nifty to see how maths can be used in epidemiology in order to make some suggestions to governments and bodies in order to try to really curb an infection. So epidemiology relies on a number of different mathematical models and metrics in order to really gauge the spread of disease. And 2 very important statistical calculations that can be used, I think the average person often conflates and confuses between, and that is the percentage difference and the percentage change. So these are not the same thing. So percentage difference examines the difference between 2 different things, whereas percentage change examines how the same thing has maybe changed from one point in time to another point in time, or an old value and a new value. They're both going to be percentages, but they don't do the same thing. So let's examine both of these using epidemiological data. So I pulled these numbers from the actual database, from the CDC database, and let's practice calculating both percentage change and percentage difference. So in this case, May 15th, 2021, the USA reported 32,534,073 COVID-19 cases, whereas Japan reported 671,841 cases on the same day. What is the percentage difference between these 2 countries in terms of COVID-19 cases? So I'm first going to subtract this value from this value. So let's actually write what's going on here. So let's call this value A and let's call this value B. So it's going to be A minus B divided by A plus B divided by 2 multiplied by 100. So let's put this in the calculator. So I got 191.9%. So the COVID-19 cases differed by 191.9% between these 2 countries. So the second example said that the USA reported 163,199 COVID-19 cases on April 1st in 2020, and then 14 days later, on April 15th, they experienced 578,268 cases. What is the percentage change? Now notice in this case that we're actually going, it's the same country, and we're looking at how did it change over time. In this case, the USA, and how did it change between April 1st and April 15th. So in this case, this would be new. This would be original. So let's do that. So I calculated that to be, now notice that percentage difference is always going to be a positive value, but percent change may actually be a negative value. If the original value is larger than the new value, then that will result in a negative percent change, whereas in this case, because the original was smaller than the new value, this will result in a positive percent change. Now you are responsible for being able to calculate both of these metrics, and you really should be familiar with their differences and how they can be used. Now, during the pandemic, for the first time, maybe, a lot of people were actually invested and interested into the work being done with vaccines and how that process works. So there was a lot of publications being done, and they were being scrutinized right away. And that's a good thing. Scrutinization is good. However, I think that people were not aware of the fact that the way that science really works is through peer review and is through replication. So as preliminary studies were being put out, people were conflating or misconstruing these findings or over-exaggerating the importance of some of these findings. So that was a big problem, and understanding that it takes time for certain studies to actually be examined, to be evaluated, to be replicated, to see, is this appropriate, is it not appropriate? And so when people are scrutinizing every single thing that's coming out before this has the chance to happen, you start to get conflicting evidence, you start to get conflicting views, and then you get people no longer trusting big science or the scientific industry as a whole, which becomes a really, really, really big problem because science is at its core founded on evidence. So a doctor is not going to prescribe a certain vaccine or a certain medicine based off their own personal opinion. They're going to do so based off of published research and actual evidence. To use an example, all vaccines, like any other drugs, have side effects. The side effects are very, very small, but they still occur. So I do remember this case still where people were going, anti-vax crowds were basically fighting against COVID-19 vaccines because people were getting anaphylaxis as a result, as a side effect. And this did happen. Anaphylaxis did occur. However, if you go to the CDC website and we go down to the data, first of all, all of these are linked publications showing the safety and efficacy of this vaccine. But if we go down to anaphylaxis, here are a number of studies showing that it is extremely rare, and the figure that they reported is 5 cases out of 1 million is where we would expect this type of side effect. So 5 out of 1 million, that is a 0.0005% chance. So that is an incredibly low figure, and this was being conflated to be like, see, vaccines are causing people death, vaccines are causing these problems. If a vaccine is being rolled out, it's only doing so after a series of tests and studies have been done to ensure the safety of this vaccine before being distributed to the public. They're not going to put out a vaccine that is not safe. Another study that I think is quite interesting is this one, where they did a meta-analysis looking at what is the role of social media on misinformation. And this study is a really, really interesting one that I would recommend you read. So these are the types of misinformation that they found regarding the COVID-19 vaccine. Conspiracy claims, so things like 5G was being put into the vaccine, or that COVID-19 was man-made, or the medical misinformation. So again, this conflating of vaccine side effects, or the vaccine itself is going to cause the disease, which is the opposite of what a vaccine will do, or even vaccine development. A lot of people are like, they're rushing this vaccine, it's not ready, or the content of the vaccines. People are like, it contains these dangerous chemicals. But again, this is really just science illiteracy, people not really understanding the science and then being easily swayed by certain figures, certain public figures, and are spreading misinformation. So I think more than ever, understanding misinformation and being able to identify misinformation is more relevant than ever. And I think that was a really big problem during the COVID-19 pandemic. I do think that this pandemic should not have claimed as many lives as it did if certain protocols were more widely accepted by the general public. Let's go over the learning outcomes for subtopic C3.2 Defense Against Disease. Recall that this only has standard level learning outcomes and there are no additional higher level learning outcomes. So the very first learning outcome that we looked at was really all to do with what exactly is a pathogen, what does this mean, and how do they cause infection. We then started to examine how does the body actually block or defend against this. And the first thing is going to be the idea of primary defense. So this learning outcome looked at the role that the skin and mucous membranes have and their role in primary defense and actually blocking pathogens from even entering the body in the first place. We then examined the blood clotting cascade process and what exactly happens when tissue is damaged and what your body does to repair this in order to stop blood from coming out, and how this also works as a means of immune defense in order to seal up a potential entry of pathogens. We then explore the difference between the innate immune system and the adaptive immune system, and how exactly these two systems are different from each other and how they're related to each other. Then looking into the innate immune system, we looked at the role that phagocytes play and actually as being first responders and what exactly they do in order to arrive to the site of infection and start to destroy incoming pathogens. We then contrasted them with respect to lymphocytes and the difference between a phagocyte and a lymphocyte, and how lymphocytes, where they're actually located, what they actually do, and different types of lymphocytes that there are. We really started to explore the idea of antigens and how antigens are really what's going to trigger an immune response, and how exactly this can lead to a person either initiating an immune response or not initiating one. And we even use blood groups as an example. We then explored how B lymphocytes and T lymphocytes work together in the adaptive immune response, and how B lymphocytes can be activated by helper T lymphocytes, and that entire process. Once B lymphocytes have been activated, we then looked at how they can multiply into plasma cells and even memory cells in order to actually retain immunity, which is what this learning outcome was really looking at, and what exactly immunity means with respect to memory cells. We then changed gears a little bit and looked at what exactly HIV is and how HIV can be transmitted from one person to another person. And note that this is not the same thing as AIDS. This second learning outcome explored how HIV infection can ultimately result in AIDS, and how these two are not the same. We then explored antibiotics and their role and function in actually curbing a bacterial infection, and why antibiotics actually work to target prokaryotes but not eukaryotes. And we then looked into how the overuse of antibiotics has actually led to antibiotic resistance in bacteria, and how this evolution is causing some medical problems and how it's being dealt with. This learning outcome started to explore zoonosis and what exactly this means, and how a pathogen can actually transfer from one host to another host, and we looked at different examples of this, including tuberculosis, rabies, Japanese encephalitis, and COVID-19. We then explored vaccines and immunization, and what exactly vaccines are, how they work, and how they take advantage of our immune system in order to build memory without the risk of actually getting the initial infection. We then explored herd immunity and how herd immunity can be used in order to prevent epidemics from actually occurring. Finally, we looked at evaluating data relating to the COVID-19 pandemic, and specifically how to calculate percentage difference and percentage change. If you wish to learn further, this page is a good place to start. So here are the different textbooks and the relevant pages that I use in order to create these lessons. It's worthwhile taking a look at some of these. I always recommend taking a look at simulators and interactives in order to further explore concepts. This one examines the immune system through kind of an interactive guide through all the different aspects of the immune system. This examines something called an ELISA test and how this can be used in diagnostics. This is a really good simulator for disease spread, and it actually allows you to tailor certain aspects of a particular pathogen or a particular disease, how vaccination can influence this. It's a really good simulator for looking at this. And this is looking at blood clotting, and it actually gamifies this entire blood cascade experience. We looked at a number of different proteins in this subtopic, and exploring them in 3D models is a really, really good avenue. All of these are links to articles where you can actually read about these proteins and also explore them in 3D space, which is always recommended to really get a better understanding of their structure and function. And finally, I listed a number of articles that I think are really relevant to this topic. Some of these refer to how vaccines work. Some of them look at breakthroughs, some of them look at COVID-19. This one right here is a really good book that I would recommend reading. It's a fantastic read. Overall, a lot of these articles are really recommended if you're really interested in the immune system in general, like I am. And finally, I would always recommend that not just to learn for the sake of a test, but learn for enjoyment, learn for the sake of learning.